Okay, welcome to video 2 in our first renal lesson. Let's talk a little bit more about how our glomerulus works in this video. So here is our glomerulus in our big kidney here. It's in this yellow box here, and of course that's where your nephron is, and this is where your Bowman's capsule and your tuft is, your glomerulus. So I'm not going to cover a lot of this anatomy. I'll leave that to the anatomists, but let's start here with the interlobular artery and vein. This branches off and it supplies blood to the individual nephrons, okay? So each individual nephron gets its blood supply from that artery, and the blood enters the renal corpuscle through the afferent arteriole, okay? Afferent, first one, it enters through that arteriole. It's going to exit through an efferent arteriole. But let's start with the afferent arteriole. The blood enters there, okay? And then it enters the glomerular capillaries. And this is like, again, it's like a ball of capillaries that you'll find that is sort of fit into that Bowman's capsule. And this is where your filtration takes place. Notice the parallel circulation, since each one of these gets its own arteries. Artery from a branch off the interlobular artery, it means that they're independent. That is, if something goes wrong with the supply to one nephron, it doesn't affect the other nephrons, and this is one of the advantages of having parallel circulation. If they were in series, then if you had one nephron that was blocked or had a problem, then it would block or it would cause a problem with all the nephrons downstream, with each one having its own little artery and individual supply. That doesn't happen. So it's kind of a safety factor. Now, and then you have your efferent arteriole. That's where your blood exits that glomerular tuft. And you enter peritubular capillaries, which wrap around the renal tubules, okay? So here's another view of that. We have two kinds of nephrons. We have cortical nephrons. These, their loop of Henle does not dip into the inner medulla like the one we're going to talk about in a minute, the juxtamedullary. So this is a loop of Henle here, and notice that this doesn't really dip too far into the medulla. It never makes it into the inner medulla. And so these cortical nephrons are really more peripheral, more in the cortex, obviously, the name cortical. And here's our juxtamedullary, and these do have very long loops of Henle that get down into that inner medulla where the osmolarity can get very high, and that's important for water reabsorption. You will note that the cortical nephrons do not have vasa recta like the juxtamedullary nephrons do. So the vasa recta is a special type of peritubular capillary that wraps around the loop of Henle, okay? And so naturally enough, there's a lot of vasa recta here because we have a very nice long loop. We do not have vasa recta associated with cortical nephrons, okay? So if we take a look at these glomerular capillaries, we see we have some branching and a lot of loops here. And the whole idea here is to maximize the surface area so that you get maximal filtration. So your blood is entering here through the afferent artery, and because you have a reasonable amount of pressure relative to the pressure in Bowman's capsule here, you actually filter plasma across the walls of this capillary into Bowman's capsule. And if you maximize the surface area, you can maximize the filtration, and so that's what all this looping is all about here. Here's another view of that renal corpuscle. We have 3 layers of filtration associated with the Bowman's capsule and the glomerular capillaries. The first, going from the capillary to the capsule, is the endothelial layer, E, okay, here. Notice that this is fenestrated. That means it's got holes in it. They're about 70 nanometers, okay? So that's our first layer that your plasma encounters as it goes from the lumen of the capillary to the lumen of the renal tubule, okay? The second layer is the glomerular basement membrane just underneath the endothelial layer. This is highly negatively charged, and that helps keep proteins out. And then we have podocytes, PO. And they form a layer of slits that roughly 8 nanometers, it varies depending on conditions, and they form a final filtration barrier in this glomerulus, or this renal corpuscle, excuse me. Notice that there are mesangial cells in here. We're not going to talk a whole lot about them in this video, but they have a function. They clean up this area and keep it free of debris. And they also can regulate filtration. They're under control of the autonomic nervous system, and that's pretty much all we're going to say about that for right now. Our filter layer 1 is fenestrated, okay? And that means that there are these holes in the endothelial layer, which is what we're talking about here, to promote filtration. And those 70 micron fenestrations are big enough to pass most proteins, most plasma proteins. Note that layers 2 and 3 are going to keep protein out, so you shouldn't find protein in your urine. But this particular layer does allow proteins through. Its primary function is to keep out really big things like the cells. Like red blood cells are kept out of the renal tubule by this layer. So if you find blood in your urine, if it's coming from the glomerulus, that's extremely bad news in terms of the integrity of this filtration barrier. Layer 2 is the glomerular basement membrane here, okay? And it primarily does its job because it's negatively charged. Most plasma proteins are negatively charged, albumin and hemoglobin, for instance. And so you have a negatively charged basement membrane, and negative charge repels negative charge, and that keeps a lot of these proteins from being filtered, certainly the bigger ones. So here is a graph. On the y-axis, we have what's called filterability. This is in other words, this is how easy a protein can get through, and this is the size of that protein in angstroms. So an 18 angstrom protein isn't going to be it's going to get through this filtration barrier. And it's going to, fairly easily. A lot of it's going to get through. But as you increase the size, less and less protein gets through, and you see you get to about this 30 nanometer point, and pretty much no proteins, or 30 angstrom point, pretty much no proteins are going to get through anything bigger than that, okay? So for instance, you'll be excluding albumin by doing that. Now, if we take the negative charge out of the filtration barrier, primarily the glomerular basement membrane, now look what happens. Our 30 angstrom protein, which wasn't getting through at all in the normal membrane, is now getting through, okay? And in fact, everything up to probably about 40 angstroms gets through. So now our albumin's getting through as well. So if you lose the negative charges on the filtration barrier, primarily the glomerular basement membrane, you're going to let a lot more protein through. And this does actually happen with some diseases, so it's not an academic point. You can lose that negative charge, and it will cause proteinuria. And if you see proteinuria, that is very bad news. Ordinarily for the urinary tract, that means something's really wrong. Ordinarily, you should see little or no protein in the urine. If you see more than a trace amount, that's probably a bad thing. There are a few things that you're going to learn to look for in clinical chemistries when it comes to the urinary system. Protein is probably going to be the first one you'll look at. If you see protein, that's bad, okay? Significant amount of protein, okay? And this is why it should keep protein out. The filtration barrier should. So do note that despite that negative charge in this filtration barrier, chloride gets through. It's relatively small. So, you're not talking about all anions. You're just talking pretty much about, the plasma proteins, particularly the larger ones, are going to be repelled by this and they're not going to get through. But you do get anions through. It's not like your urine is going to be completely deficient in things like chloride, okay? So you should recognize that. And then finally we have our third layer, which is our podocytes. These are epithelial cells associated with the Bowman's capsule now, Okay, and they're your last filtration barrier. They have or form foot processes that interdigitate, and they have slits in between them that allow things to get through. They're about 8 nanometers, and so this is your last filtration barrier, your last line of defense in terms of keeping things like proteins out. Okay. And I think I'm going to stop there, and let's move on to video 3, where we'll talk a little bit more about the process of urine formation and some of the things that go into that. Thanks.